Hello everyone, a very welcome to Vyas Edification Kota. So today in the series of NCERT discussion, we are going to talk about a very interesting chapter, an important chapter, that is Anatomy of Flowering Plant. We are going to discuss each and every question of NCERT textbook in this session. This chapter is from class 11th and from unit 2. And we are going to discuss each and every question of it in this series. This chapter contains total 12 questions. So let's begin with this. So very first question we have on the screen, that is state the location and functions of different types of meristems. So we have to state the location as well as the functions of different types of meristems. So basically we have three types of meristems, that is apical meristem, intercalary meristem and lateral meristem. Now if we talk about the very first type of meristem, that is apical meristem, then apical meristem is found on the apex or apices of stem, roots and their branches. What is the location of apical meristem? They found on the apices of root, stem and their branches, or apex of root, stem and their branches. This is the location of apical meristem. Now, what is the function of apical meristem? It helps in the growth. It helps in the growth in length and also forms primary tissue. So this is the function of apical meristem, that all the primary tissue is formed with the help of this apical meristem, and also the contribution in the length of a plant is coming from apical meristem. Then the second type of meristem we have is intercalary meristem. Now, where it is found? Intercalary meristem is found on above and below of stem nodes and also on the leaf bases. So what is the location of intercalary meristem? It is found above, just above and below of stem nodes and on leaf bases. Right? Now what is the function of this intercalary meristem? It also helps in the growth of internode. It helps in the growth of internode and also helps in the growth of leaves. So this is the function of intercalary meristem. Then the last one we have is lateral meristem. Now this lateral meristem is of two types, that is cork cambium and vascular cambium. Cork cambium and vascular cambium are the two types of lateral meristem. So let's discuss the first one, that is cork cambium, which is also known as phallogen. Now cork cambium or phallogen is actually formed from the hypodermis layer of stem, and if we talk about in case of roots, then this cork cambium or phallogen is formed from the pericycle. So this is the location or formation of cork cambium or phallogen, which is a type of lateral meristem. Cork cambium or phallogen, I am repeating it, cork cambium, which is also known as phallogen, is going to form from the hypodermis layer of stem and pericycle in roots. Now let's discuss the function of this cork cambium or phallogen. So this cork cambium or phallogen is going to form a protective layer of cork, a protective cork and aerating lenticels on the outside. It is going to form aerating lenticels and a protective cork layer on the outside and another layer in the inner side, which is also known as phalloderm. Now the outer one protective cork layer is known as phallem, right? Now another type of lateral meristem we have is vascular cambium. If we talk about vascular cambium, which is another type of lateral meristem, then vascular cambium forms from the intrafascicular cambial rings in stems, intrafascicular cambial rings as well as interfascicular cambial rings or strips are helping to form these vascular cambiums in stems. If we talk about the formation of these vascular cambium in roots, then they are going to form from parenchymatous tissue and pericycle, right? So where this vascular cambium is actually forming and where do these actually found? So this vascular cambium in stem is found or formed from intrafascicular cambial strips as well as interfascicular strips. Now, in roots, it is formed from parenchymatous tissue as well as from pericycle. Now, what is the function of this vascular cambium? It helps the formation of secondary phloem. This vascular cambium, which is a type of lateral meristem, is going to form secondary phloem on the outer side and secondary xylem on the inner side. So this is the function of vascular cambium, and with this we have completed the explanation of this question number 1. Now we are going to write the answer of this question. So first we'll write start like this. We have three types of meristems. We have three types of meristems. Number 1 we have is apical meristem. Apical meristem. Now, this apical meristem, we know that it is found on the apex of stem, root and their branches. So we'll write this. These found on apex of root, stem and branches. So this is the location of apical meristem. Now if we talk about the function of this, then we know it helps in the, if we talk about function, if we write about function, then it helps in growth in terms of length, also it forms primary tissue. So this is the function, or this is the location, function as well as location of apical meristem. Let's come towards the next meristem, that is intercalary meristem. Intercalary meristem. This is the second type of meristem we have, and it is found above, just above and below stem nodes and on the basis of leaves. So intercalary meristem is found just above and below stem nodes and on leaf basis, Now what is the function of intercalary meristem? We know that if we discuss the function, then we know it also helps in the growth of internodes, which is going to eventually help in the length or growth in the terms of length of a plant, right? So function of intercalary meristem is it helps in the growth of internodes and growth of leaves, right? Then next and the third type of meristem we have is lateral meristem, which is of two types, that is cork cambium and vascular cambium. So the third one we have is lateral meristem. Lateral meristem, in which we have A, that is cork cambium, and this is formed from hypodermis layer in stem and from pericycle in roots. If we talk about the function, of this, then this lateral meristem is going to form a protective cork and lenticels, aerating lenticels on the outer side. So the function of this cork cambium, which is also known as phalogen, is going to form protective cork plus aerating lenticels on the outside and phalloderm on the inner side, right? So this is the function and location of cork cambium, which is a type of lateral meristem. Then the last one we have is under this one only, the lateral meristem, which is having another type of lateral meristem, that is B. We have vascular cambium. We know that vascular cambium forms, if we talk about vascular cambium, then it forms from intrafascicular cambial strips and interfascicular strips in stem and from pericycle in roots. Parenchymatous tissue as well as pericycle in roots. So this is formed from this kind of tissues, such as intrafascicular cambial strips and interfascicular cambial strips in stems, and in root it develops from pericycle region. Now what is the function of this vascular cambium, which is a type of lateral meristem? It forms secondary phloem on the outside and secondary xylem on the inner side. So if we talk about function, then it forms secondary phloem on the outside and secondary xylem on inner side, right? So this is the complete solution of question number 1, in which we have already covered all 3 types of meristems we have, that is apical meristem, intercalary meristem and lateral meristem. With this we have completed the solution number 1. So now we have question number 2 on the screen, that is cork cambium forms tissues that forms the cork. Do you agree with this statement? If yes, then explain. Yes, we totally do agree with the statement. That statement is cork cambium forms tissues that forms the cork. How? Let's understand. First of all, when the secondary growth takes place in dicot stem and roots, whenever the secondary growth takes place in case of dicot root and stems, which is not going to take place in monocot stem and roots. So during the growth or during the secondary growth in dicot stem and roots, the epidermal layer in these dicot root and stem gets broken down. During the secondary growth in dicot stem and roots, the epidermal layer gets broken down. So to replace these epidermal cells, cork cambium develops there to provide protection to the stem and roots from infections. All right, to replace all these epidermal broken cells, for providing protection to the stem and roots from infection, a new thing which is known as cork cambium develops from the cortical region. This cork cambium is also known as phalogen, and this layer contains or is composed of thin-walled rectangular cells. Cork cambium is also known as phalogen. Now, what does it do? First of all, this cork cambium have bipolar division. Now, what does that mean? This means that this cork cambium cuts off the cells on both the sides. This cork cambium is going to form the cells on both sides, that is on outer side and on the inner side. Now the cells present on the outer side is going to form a cork layer or a protective layer of cork, which is also known as phloem. While the cells present on the inner side is going to form another type of tissue, that is secondary cortex, which is also known as phyloderm. Now this cork cambium is actually impervious to water, but it allows gaseous exchange through lenticels. Lenticels are also present on the outside. Now all these layers such as phylogen, phloem and also your phyloderm, all these three layers constitute together to form a new layer known as periderm. So this is all about the explanation of question number 2, and now we are going to write answer number 2 on the screen. So here, let's start writing answer. Yes, we totally do agree. First of all, we write yes, we do agree with the statement, Then we have to explain why do we agree with the given statement. So answer will go like this: when the secondary growth takes place in dicot, root and stems, the epidermal layer gets broken down, So to replace the epidermal cells, there is a need to form cork cambium to provide protection. Why this cork cambium is actually developing or forming? To provide protection to stem and root from infection. Right? Why the cork cambium is getting developed? This is going to form to provide protection to stem and roots from infection, because during the secondary growth in dicot, stem and roots, the epidermal layer gets broken down. All the cells in the epidermal layer gets destroyed. That is why, to provide protection to the stem and root of dicot plants, there is a need to form cork cambium, a special kind of tissue which is providing protection to them. Right? So this is the purpose. Now, what does this cork cambium actually do? This cork cambium have a special function, that is bipolar division. Bipolar division is present in it. What does that mean? That means it cuts off the cells on both sides. Now on the outer side, the cells are differentiated into protective cork known as phloem, while the cells on inner side forms secondary cortex known as falloderm. Now, this cork cambium cuts off all the cells on both the sides. The outer cells are going to differentiate into this layer which is known as phloem, which is protective in nature, which is going to protect the stems and roots. And on the inner side, which are the cells which are present on the inner side, are going to form secondary cortex, which is also known as falloderm. Alright, now this cork cambium is actually impervious, It does not allow water transport. So it is impervious to water, but allows gaseous exchange through lenticels, right? Now, this phloem plus phloem and falloderm, all these constitute to form a new layer which is known as periderm. So all these constitute to form periderm, or we can call this layer which is constituting all these three layers such as phloem, and falloderm on the inner side. The inner side one is falloderm and on the outer side it is phloem. Right, the phloem. So all these three layers together are forming a new layer which is known as periderm. So with this we have completed the solution number 2. Now let's talk about the next question, that is question number 3 on the screen. Explain the process of secondary growth in the stems of woody angiosperms with the help of schematic diagrams. What is its significance? So for this question, first of all, the process, we have to explain the process of secondary growth in the stems of woody angiosperms. So as we know that the process of secondary growth in the woody angiosperms takes place with the help of two types of cambia, that is cork cambium and vascular cambium. So moving on to the explanation of this answer, we have to draw the schematic diagrams of this. So first of all, we are going to draw the vascular cambium here, vascular cambium, and here we are going to make schematic diagram of cork cambium. Alright, so let's start to draw schematic diagram for vascular cambium. So this is your epidermis layer, epidermal layer or epidermis layer. Then you have cortex region, and inside this you will see vascular bundles like this. Secondary growth always occurs in dicot plants only. Secondary growth is not the feature of monocot plants. We know that. So, secondary growth occurs in dicot plants, in angiospermic dicots, and we are talking about the process of secondary growth in angiospermic dicots in stems, right? So this process or this secondary growth in angiospermic stems takes place with the help of two types of cambia, cork cambia and vascular cambia, and we are writing or drawing the schematic diagram for vascular cambium here. So this is your epidermis layer. Let's label this epidermis. Then we have cortex region. So this is your cortex. Now these are your vascular bundles in which you will find the primary xylem and primary phloem. So on the outer region you are having primary phloem, I'm writing the primary like this, primary phloem, and on the inner side this you are having primary xylem. Now you will see the rings between primary xylem and phloem in these bundles like this. There are rings present between primary xylem and phloem. These are cambium rings or cork cambium. So we'll label this as cork cambium. The innermost region here you can see is pith, is known as pith. And the region or the cells between all these vascular bundles, you will call them as medullary rays. So these are medullary rays. Medullary rays. Right? So this is the schematic diagram of a dicot stem which is showing you vascular cambium or showing you the secondary growth in the angiospermic dicot stem via vascular cambium. Now let's draw another type of cambium, that is cork cambium. Now you will see cork cambium we are going to make. So this is your innermost circle. Bundles are present, then I'm talking about the secondary growth happened. I'm drawing the diagram or schematic diagram when the secondary growth is being taken place. So this is your circle having bundles, then this is your secondary xylem, then this is your phloem, then the secondary phloem on the outer side, secondary phloem on the outer side, secondary xylem on the inner side. Then you have a layer, protective layers of cork which is known as phallogen, and the outermost protective layer you have is phallem. This is you are having phallem. Right? Now this is your phallem. Let's label these. This is phallem. This is phallem. Then the second layer we have is phallogen. Phallogen. This is the third layer is your secondary phloem. Secondary phloem. Then you have this as secondary xylem. Now you are going to make rings, cambial ring and medullary rays. So these are medullary rays like this. These are medullary rays. These are medullary rays. So let's write first of all this is as medullary rays, right? And this is your cambial ring. This is your cambial ring. Here we have is cambial ring. Cambial ring. Right, so we have drawn the schematic diagrams for cork cambium and vascular cambium as well. Now, we know that the process of secondary growth in the stem of woody angiosperms takes place with the help of two types of cambias, that is vascular cambium and cork cambium. We know that. So let's discuss how actually the secondary growth in dicot stem takes place. First of all, if we talk about with the help of vascular cambium, then we know that in this diagram you can clearly see that these are the cambial strips. These are the cambial strips which are present in between primary xylem and primary phloem. These strips in dicot stem during the process of secondary growth, which is present between primary phloem and primary xylem, is actually known as cambial strips, and the name of these cambial strips which is present between primary xylem and primary phloem is known as intrafascicular cambium, intrafascicular strips. Now, here you can see the middle portion or the cells which is present between the bundles are known as medullary rays. So these cells of medullary rays are going to form another strip between these bundles here. So these strips formed from the cells of medullary rays are going to form another kind of strip that is known as interfascicular strip. So new strip is going to form like this, new strip is going to form like this from the cells of medullary rays, and this interfascicular strip formed from the cells of medullary rays adjoining the intrafascicular strip which is present between the primary xylem and phloem. So this intermixing or adjoining of interfascicular strip and intrafascicular strip makes a new ring which is known as cambial ring. Now this cambium ring or cambial ring is going to form new cells on either sides, that is on the inner side and outer side. This cambial ring have bipolar division. This ring cuts off new cells on both the sides. The cells on the outer side here are going to convert into secondary phloem, and the cells present towards the pith are going to differentiate into secondary xylem. So this is how the secondary growth takes place in dicot stem with the help of vascular cambium. Now if we talk about the another type, that is we have cork cambium, then we know that cork cambium produces secondary growth, and all this secondary growth tissues, cork cambium produces secondary growth tissues, and all these tissues are known as peridome. The tissues formed from the cork cambium which is helpful in the secondary growth are collectively called as peridome. Now this cork cambium is also known as phallogen, is produced secondarily from the epidermal cells of living tissue. Right? Now what happens in cork cambium? In this cork cambium, the cambial strips are going to form cork on outer side which is known as phallum, and inner cortex or secondary cortex on the inner side which is known as phalloderm. So this is how the secondary growth in dicot stem takes place with the help of cork cambium. This cork cambium is going to produce secondary tissues in the stem with the help of forming of cork on the outer side and secondary cortex which is also known as phalloderm on the inner side. Right? Now the outer side of this, the outer side of cork which is also known as phallum. Phallum is also having lenticels or aerating pores on it. These lenticels are helpful in the gaseous exchange. So this is how the secondary growth in dicot stems takes place. Now let's write answer for this question. We are writing like this. Answer number 3. The process of secondary growth. The process of secondary growth takes place with the help of two types of cambia. The one we have is cork cambium and second one we have is vascular cambium. So first of all, let's write that is vascular cambium. First we are going to write vascular cambium. Now vascular cambium, in vascular cambium we'll write like this. In woody stems, what we see, the strips present between the woody strips present between the primary xylem and phloem is known as intrafascicular ring or strip. Now, the cells of medullary rays, forms the interfascicular ring between the vascular bundles which adjoining the intrafascicular ring. With adjoining interfascicular rings formed from the medullary cells is adjoining the intrafascicular ring. This results into the formation. Of cambial ring. Now, after the formation of cambial ring, this divides the cells. This differentiates or cuts off the cells on either sides, right? So this cambial ring cuts off the new cells. That means it forms new cells on either sides. On the outer side, it forms the secondary phloem. And on the inner side, it forms secondary xylem. Remember that secondary xylem always produces a large amount as compared to secondary phloem. So we'll write like this: secondary xylem. Always produces in large amount as compared to secondary phloem. So this is about vascular cambium, or the secondary growth happens in dicot stem with the help of vascular cambium. Now we are left with the secondary growth in dicot stem with the help of cork cambium. Let's write that also. So let's write cork cambium. Now we know that this produces secondary growth tissues which are collectively called periderm. So this this cork cambium produces secondary growth tissues, and these secondary growth tissues all together or collectively are known as periderm. We know that. Now this cork cambium is also known as, it is also known as phallogen, and it produces secondarily from the supraepidermal, that means the outermost epidermal layer. This cork cambium produced from the outermost epidermal layer, epidermal cells of living tissue. Right? Now what happens in the secondary growth, or how this cork cambium helps in the secondary growth in dicot stems? So it produces the cork or phallum, because this cork is also known as phallum on the outer side, and produces the secondary cortex, also known as phalloderm on the inner side, right? Also on the outer side of this cork cambium, that means on phallum, they're having aerating pores known as lenticels, which are helping them in gaseous exchange. So on phallum there is lenticels helping in gaseous exchange. So this is all about the secondary growth in dicot stems of angiosperms with the help of these two types of cambiums, that is vesicular cambium and cork cambium. Now in this question we are left with the significance of the secondary growth. So let's write the significance of the secondary growth. We are going to write significance. So the first one is that it replaces an old non-functional old tissues with active new tissues, right? Second one is the plants who have secondary growth in them are living longer. They live longer as compared to other plants. So the plants having secondary growth live longer as compared to other plants who are not having secondary growth, right? Then, all right, we have this two significance. The third one we have is this commercial cork is the product. The commercial cork we are getting from the market is actually the product of this secondary growth only. So this commercial cork is the product of secondary growth. Also the wood we get is also the product of the secondary growth. So this wood is an important product of secondary growth, right? So with this, we have completed the whole answer of question number 3, with along with the secondary growth, how the secondary growth takes place in dicot stems of angiosperms, along with its significance. So this is the complete solution of question number 3. So now we are going to discuss the next question, that is question number 4 of this chapter, and here we have to draw some diagrams. Let's read the question number 4. Draw illustrations to bring out the anatomical difference between monocot root and dicot root. Then we have to draw a diagram to illustrate the difference between monocot stem and dicot stem. As you can see on the screen, I have bought diagrams which are already made by myself only. Now we are going to label all these diagrams. So in this, you can see here on the screen, we have 2 diagrams, and these 2 diagrams are of monocot root and dicot root. So basically this one diagram is representing the monocot root, whereas this diagram on the right side is representing the dicot root. Now let's just label it. So now we are going to start labeling it. The first one, we are starting it from outer to inner side. The first one structure, you can see the hair-like structure. The hair-like structure in the root you are finding is actually root hair. So these are root hairs. Obviously root hairs are going to find on roots only. In roots only, they are not going to find in stems. So root hairs are present in roots. So you can see these structures. These structures are nothing but root hairs. Right? Now let's talk about the first layer on which these root hairs are actually attached. So this layer is known as epidermis. It is epidermis. Now moving on to the next labeling, here this tissue, this whole tissue here, this tissue is known as cortex. This is known as cortex. Then if we talk about this one structure in the middle, you will see this is metaxylem. So this is metaxylem. Then again, these structures you can see is phloem. These structures are phloem. So these are phloem. These are phloem in monocot roots, right? And this is your conjunctive tissue. This is your conjunctive tissue. And this whole diagram is actually representing the monocot root. So we can write here also, this is the diagram representing monocot root. Now let's come towards the next diagram, that is we have a diagram of dicot root. Now we are going to label it here. So first thing you can see, always the hair-like structures in root diagram or anatomy of root diagram, you will find root hair. So also these are root hairs, same as the monocot root. Then this is your epidermis layer. This is your epidermis. Then you have of course cortex layer here, cortex. Then you will see this layer is endodermis. This outermost layer here you can see is endodermis. Endodermis. Next to endodermis you will find another layer which is known as pericycle. So this is your pericycle. This is pericycle, right? Now these are phloem. These are phloem, and this is your protoxylem. Protoxylem. And the innermost, this one is representing metaxylem. Metaxylem. One thing you have to remember is we have 2 types of xylem, protoxylem and metaxylem. In case of roots, protoxylem will be at the outer side and metaxylem will be at inner side. And if you talk about stems, then protoxylem will be at inner side and metaxylem will be at outer side. This is the basic difference between these. Now we have labeled with both these diagrams of monocot root and dicot root. Now we are moving on to the next diagram which is depicting monocot stem and dicot stem. Here you can see this is the structure, this is the diagrams. Both the diagrams are of stems, are of anatomy of stems, monocot and dicot. This one is representing the stem of monocot and this one diagram is representing the dicot stem. Both are the stems diagram. So let's try to label. First of all, here also in these diagrams you will find hair-like structures. Now, you cannot call them as root hair because root hairs are only present in roots, of course. Then, what are these hair-like structures which are present on stem are called? These are trichomes. These are trichomes. So, we will write trichomes here. These are trichomes. Similarly, here also we will write trichomes. These are also trichomes here. Then, the first layer, extra layer present in the stem is cuticle. You must have heard about cuticle. This layer is cuticle. This layer is cuticle. Then next to cuticle, we have epidermis. We have as epidermis. So both these here in this diagram, this one layer, and in this diagram, this one layer is representing epidermal layer, epidermis. Outer to epidermis, there is presence of cuticle and extra layer, and also in the stem, we have hair-like structure which are known as trichomes, right? Then if you talk about these tissues, this is chlorenchyma, right? Now, here also in the monocot stem, you will find the vascular bundles are present in a scattered way, in a scattered arrangement. So these bundles in dark color is actually representing vascular bundles, and all these are the ground tissues. Except for these tissues, we have ground tissue, and of course here somewhere we'll have pith, right? So this is the labeling of monocot stem. Now let's talk about the labeling of dicot stem. In the dicot stem also we have trichomes which are hair-like structure present in the stem. Then we have cuticle also in this. So the first layer here is actually cuticle. This is also cuticle in dicot stem. Then the second layer next to cuticle is epidermis. Here epidermis, this is also epidermis. Then we have this layer. As collenchyma. This one layer is chlorenchyma. Then we have a layer of parenchyma, which is a living tissue which stores food, of course. Then we have endodermis. Here this layer is representing endodermis, and these are actually representing vascular bundles in dicot stem, which is present in a ring manner or in a ring arrangement. So these are vascular bundles. I'm writing VB, vascular bundles. Then this is your pith. Alright, so we have already labeled the monocot and dicot roots in the previous slide, and in this slide we have labeled the monocot and dicot stem. So this is how you will make the diagrams in your board exams, and with this we have completed the solution for this question. Now we'll be talking about question number 5, and the question number 5 is asking, cut a transverse section of young stem of a plant from your school garden and observe it under the microscope. How would you ascertain whether it is a monocot stem or it is a dicot stem? And we have to give reasons. So we will have to take a sample of stem, and we have to identify whether the sample of stem we have taken is a dicot stem or a monocot stem. So obviously any sample can be identified on the basis of some characteristics. Alright, so if we are talking about the stem, if we talk about the stem of any plant as a sample, we'll take the sample and identify the sample of stem, whether it is a dicot stem or monocot stem, depending upon or on the basis of characteristics we know about the dicot stem and monocot stem. So first of all, I'm going to tell you about characteristics of dicot stem, right? On the basis of these characteristics only, we will identify the stem sample is actually dicot or monocot in nature. So here we have the characteristics of dicot stem. First of all, the dicot stems, if you talk about the dicot stems, they have vascular bundles, alright. They have vascular bundles which are arranged in a ring manner. First of all, the vascular bundles are arranged in a ring manner around centrally located pith, right? The next, if we talk about, then the vascular bundles in dicot stems are conjoint, they are collateral, and they are open. That means you will find secondary growth in dicot stems, which is not found in monocot stem. Alright, this is also a characteristic of dicot stem. What I've said, that in dicot stems, the vascular bundles are conjoint, collateral, and open. That means they can have secondary growth in them. Also, they have a strip, centrally located cambium strip, which is present in between xylem and phloem, right? The second characteristic I have told about is the vascular bundles in dicot stems are present in a ring manner, in a ring arrangement around centrally located pith. The third characteristic of dicot stem is they have ground tissue. Yes, they have ground tissue. The ground tissue of dicot stem is differentiated into collenchyma, parenchyma, endodermis, and endodermis, pericycle, pith, etc. Right? So all these are the characteristics of dicot stem. Now, if we talk about the characteristics of monocot stem, what are the characteristics? The characteristic of monocot stem are as follows. There will be present an extra layer known as hypodermis. Hypodermis you will find in monocot stem only. This hypodermis layer of monocot stem is made up of sclerenchyma cells, right? Then if we talk about vascular bundles in monocot stem, then vascular bundle in monocot stems are conjoint. They are actually closed. That means there is no secondary growth is present in monocot stems. Also, if we talk about the arrangement of vascular bundles in monocot stem, they are arranged in a scattered manner. They are not arranged in a fixed pattern just like dicot stem. They are present in a scattered manner. Right? All these are the characteristics by which we can clearly identify that whether the sample we have taken of a stem is dicot stem or monocot stem. So let's write answer for this question. Answer 5. Okay. Answer 5 will be like this. The unknown sample of stem can be identified on the basis of characteristics of monocot and dicot stem. For this, or to identify the sample, unknown sample, we have to know the characteristics of dicot stem as well as the characteristics of monocot stem. So we will write the characteristics of monocot and dicot stem over here. In the left column, we are going to write the characteristics of dicot stem, and in the right column, we are going to write the characteristics of monocot stem. So let's write the first characteristic, that is the vascular bundles. Vascular bundles of dicots are conjoint, collateral and open. That means they can have secondary growth along with the cambium strip present in between xylem and phloem. Then the second characteristic of dicot stem is vascular bundles are in ring manner. They are arranged in a ring manner around pith, around centrally located pith, right? And the ground tissue in dicot stem is differentiated. Ground tissue is differentiated into collenchyma, parenchyma, pericycle, pith, etc. Right? These are the characteristics. These are some characteristics of dicot roots on the basis of which we can identify whether the sample we have taken is a dicot stem or not. Right? We are talking about stem, right? Then if we talk about the characteristics of monocot stem, then you will be finding a hypodermis layer, which is made up of sclerenchyma. Then second of, if we talk about, they have vascular bundles. Yes, they have vascular bundles which are conjoined and closed and scattered. So they have vascular bundles which are conjoined and closed. That means there is no secondary growth is found in the monocot stems, and the vascular bundles are scattered. Right? So these are the characteristics, some characteristics of monocot stems. We are discussing the characteristics of monocot and dicot stems, on the basis of which we can identify the unknown sample, whether the sample is monocot stem or a dicot stem, right? So with this we have completed the solution of question number 5. Okay, now let's discuss question number 6 here. The transverse section of a plant material shows the following anatomical features. A. The vascular bundles are conjoined, scattered and surrounded by a sclerenchymatous bundle sheets. The B we have given as phloem parenchyma is absent. What you will identify it as? So first of all, you have to know, that if you're talking about monocot stem, then in monocot stem you will find vascular bundles. Yes, of course there are vascular bundles which are conjoined, collateral but are closed. That means there is no secondary growth is found in monocot stem. Whether the secondary growth is found in dicot stems, right? So in monocot stems you will find the vascular bundles are closed, not open. Vascular bundles are open in dicot stems. Whether vascular bundles are closed in case of monocot stems. Vascular bundles are conjoint and collateral in both, but vascular bundles are closed in monocot stems and are open in dicot stems. Then moving on to the next, that is vascular bundles are actually surrounded by a sclerenchymatous tissue known as bundle sheath. So you will find this feature in monocot stems only and not in dicot stem. Vascular bundles are conjoint, collateral and open in case of dicot stems, and in case of monocot stem you will find conjoint, collateral, closed vascular bundles which are surrounded by a sclerenchymatous bundle sheath, right? Also you will find no parenchyma is present in monocot stem. There is no parenchyma present in monocot stem. And also if you compare dicot stem and monocot stem, you will find medullary rays. Medullary rays, or the tissue present between xylem and phloem, the tissue present between vascular bundles in dicot stem. Whereas in monocot stem, you will not find medullary rays or the tissues present in the vascular bundles, present between the vascular bundles, because vascular bundles in monocot stems are not arranged in a fixed pattern. They are scattered in the ground tissue. They are scattered in the ground tissue. This is the very basic difference between monocot and dicot stem, that the vascular bundles in monocot stem are not arranged in a proper manner. They are scattered in ground tissues, whereas they are properly arranged in a ring manner or a ring arrangement in dicot stems. The second most important thing is, in dicot stem, the vascular bundles are open. That means they can have secondary growth. And in monocot stem, there is no secondary growth you will find, right? So the vascular bundles are closed. Also, there are no medullary rays present in monocot stem. So in the question you can see we have given some material with features. We have given a material or a sample, you can call it, with along with some features, and the features are mentioned in the question on screen. That features are the vascular bundles are conjoint and scattered, and also they are surrounded with a sclerenchymatous tissue which is known as bundle sheath, and also the sample or the material you have given is actually absent in phloem. There is no phloem present in the material. So from this feature you can easily identify that the given material is monocot stem, right? So we will write the answer like this: the material shown feature like vascular bundle is conjoint and scattered conjoined and scattered and surrounded by a sclerenchymatous bundle sheath, and surrounded by sclerenchymatous bundle sheath. Right? So this kind of feature you will find in monocot stems only, obviously, and also the monocot stems are devoid of parenchyma and medullary rays From all these features, you can identify this material as monocot stem. So clearly, hence, from the features mentioned above is showing the material is monocot stem. right? So clearly you can identify the stem, identify the sample along with the features given on the screen. You can identify that sample is actually monocot stem. Along with this, we have completed question number 6. Now let's move on to the next question, that is question number 7 on the screen. Here we have, why are xylem and phloem called as complex tissues? Right, so to answer this, we know that tissues are made up of cells, right? When cells group together, they form tissue. Alright, but the question is asking that xylem and phloem are known as complex tissue. Why they are known as complex tissue? Obviously, we know that when the cells group together, they form tissue. But why xylem and phloem are known as complex tissues? So the answer of this question is, xylem and phloem are known as complex tissues because they are made up of different kind of cells. And the arrangement of different kind of cells together to form a tissue known as xylem and phloem, that is why they are known as complex tissues, because they are made up of different kind of cells. Now all these different kind of cells which are making up xylem and phloem tissues as a complex tissue, all these different cells are working together in a coordinated manner and as a unit to perform various kind of functions. Xylem tissue and phloem tissue are two different tissues and are totally different from each other, but present in a plant. Xylem conducts water from roots to leaves, whereas phloem tissue conducts food or transports food from leaves to other parts of a plant. So this is why the xylem and phloem known as complex tissue. Answer is simple, that these are made up of different kind of cells. So let's write answer of question number 7. So xylem and phloem are made up of different kind of tissues, right? These different not tissues, here we are going to write cell. The appropriate word is cell, because tissue are made up of cells, not tissues are made up of tissue. So here we can write different kind of cells, right? So xylem and phloem are made up of different kind of cells. Different kind of cells group together to form a tissue, and this makes them as complex tissue. So these different cells in the complex tissue, works together in a coordinated manner as a unit to perform various functions, right? These different kind of cells which are making up complex tissue known as xylem and phloem makes them as complex, right? Because in general what happens when we are talking about tissue, the tissue is actually the group of cell. But to make up a tissue, all similar kind of cells are grouping together to form a tissue. But in case of xylem and phloem, different kind of cells group together to form these tissues. That is why we call them as complex tissues. Now these complex tissues are having different kind of cells. All these different kind of cells work together in a coordinated manner and as a unit to perform various functions. Now xylem is different. Of course it is different from phloem, as xylem conducts water It conducts water from roots to leaves, whereas the phloem, we know that. What is the function of phloem? Phloem is a special kind of tissue which transports food from leaves to other parts of plant, right? Other parts of plant. That is why the transportation of xylem or the transportation of water with the help of xylem is said to be unidirectional, that is from root to leaves, and the transport of food materials such as sucrose from leaves to other parts of a plant with the help of phloem is said to be bidirectional, right? So with this we have completed the solution of question number 7. Now let's talk about the next question, and the next question we have is question number 8, which is asking what is stomatal apparatus? Explain the structure of stomata with a labeled diagram. First of all, we have to tell what is stomatal apparatus. So stomatal apparatus contains a pair of guard cells with or without subsidiary cells. Stomatal apparatus is actually a apparatus which is containing a pair of guard cells, sometimes with subsidiary cells and sometimes without subsidiary cells. So we will write the first part of this question like this: stomatal apparatus. Stomatal apparatus. Also we are going to draw the diagram of stomata, but first of all we are going to write the first part of this question, that is stomatal apparatus. What is stomatal apparatus? Stomatal apparatus is consisting of pair of guard cell with or without subsidiary cells. So stomatal apparatus is not a new thing. Stomatal apparatus is actually when we call when we have pair of guard cells along with subsidiary cells. Subsidiary cells can be present or can be absent, right? So any apparatus which is having a pair of guard cell or along with subsidiary cell, we'll call it as stomatal apparatus. Now we have to explain the structure of stomata and we have to label it as well. So first of all we are going to make the structure of stomata. So here I'm going to draw structure of stomata like this. These are the guard cells, these are chloroplast, these are the subsidiary cells, and these are epidermal cells. These are epidermal cells. Now let's mark it. So these are epidermal cells. These are epidermal cells, right? So these are, second one is subsidiary cells. Subsidiary cells are the cells which are surrounding guard cells, right? So along the cells which are surrounding guard cells are known as subsidiary cells. So these are subsidiary cells. Then these two cells which are present in a bean or kidney shaped, here it is actually bean shaped. These are guard cells. A pair of guard cells we have. So this is guard cell. This is stomatal pore. This is stomatal pore. Right? We have completed the labeling of this. Also, we can write this is chloroplast, which is present in guard cells, of course. Chloroplast. Now, we have another kind of stomata also, which is present in grasses. So, we are going to make here the diagram of stomata, which is present or which is found in grasses, and the guard cells in grasses, the guard cells of stomata in the grasses is found to be dumbbell shaped. It is bean shaped, but in grasses, the guard cell, it is dumbbell shaped. So, we are going to make stomata. This is guard cell. This is another guard cell as it is present in a pair. Right. Then here also they are also having subsidiary cells, and these are epidermal cells. These are epidermal cells. So let's write it. These are epidermal cells. Right? These are epidermal cells. Then these are subsidiary cells. These two are subsidiary cells. These two are dumbbell shaped. As you can see, these are dumbbell shaped. Dumbbell shaped guard cells are present in grasses. Dumbbell shaped. So these are dumbbell shaped guard cells. Guard cells, and these are subsidiary cells, of course. These are guard cells, and the pore in between is stomatal pore. Stomatal pore. Right? This is stomatal pore. So now we know that what is stomatal apparatus. It is actually having or consisting of a pair of guard cell along with or without subsidiary cells. This is a stomatal apparatus. We have drawn the diagram of stomata, and now we have to explain the structure. Now we have to explain the structure. So let's see. First of all, if I talk about the guard cell, there is a pair of guard cell, right? Now, guard cell is bean shaped. Guard cell is bean shaped in general and is having chloroplast and small vacuoles in it. Right? So, guard cell is having chloroplast and small vacuoles in it, and they are bean shaped, but they are dumbbell shaped in grasses. The guard cells are dumbbell shaped in grasses. Right? Now, let's see how they create a pore. First of all, the function, if we talk about stomata or stomatal pore, then they functions as, or they provide the site for gaseous exchange and also helps in the transpiration, right? So the stomatal pore is actually the site where transpiration takes place, and through this also the another process known as gaseous exchange also takes place. Now what happens? This stomatal pore closes and opens depending upon the condition. But how does it happen? So you will see guard cell have two layers, thick layer and thin layer, thick walled layers and thin walled layers. So this layer of guard cell is known as thin layer, thin walled layer, and this portion of guard cell, inner portion, which is present near the pore, you can see, is thick walled. This layer is thick walled. This layer is thick walled. In both the cases, the layer of guard cell, one layer is thick walled and one layer is thin walled. So what happens during the endoosmosis in guard cell, this thin layer, thin walled layer expands and moves outwardly. As a result, this thick wall of guard cell also moves outwardly and creates a pore in between. This is how a pore or stomatal pore is created. So this is how what happens, or this is actually a structure, and the detail or the explanation of structure we have already discussed. Now we have question number 9 on the screen. Name the 3 basic tissue systems in the flowering plant. Give the tissue names under each system. So we have to tell about all the 3 basic tissue system present in flowering plant. As we know that we have basically 3 tissue system present in flowering plant, that is epidermal tissue system, ground tissue system and vascular tissue system. Now we have to tell what tissues are included in each of these tissue systems of flowering plants. So let's write or let's discuss. First of all, if we talk about the epidermal tissue system, then epidermal tissue system of flowering plant contains epidermis, cuticle, hairs, trichomes and stomata. Yes, stomata is included in the epidermal tissue system of a flowering plant. Then if we talk about the next tissue system, that is ground tissue system, then ground tissue system of plant or of a flowering plant includes collenchyma, parenchyma, sclerenchyma and mesophyll cells. The last one we have is vascular tissue system. Vascular tissue system of a flowering plant contains xylem, phloem and cambium. So this is all about this question, and now we are going to write the answer of question number 9. So basically we have 3 tissue systems present in the flowering plants, such as the first one we have is epidermal tissue system, epidermal tissue system. Then second one we have is ground tissue system, ground tissue system, and the last one we have is vascular tissue system. Now we are going to write the tissues included in these tissue systems. So first of all, if we talk about the very first one, that is the epidermal tissue system. What are the tissues included in epidermal tissue system? That is epidermis. Epidermis is included in epidermal tissue system. Then trichomes of stem. Then hairs, root hairs. Then stomata is also included in epidermal tissue system. If we talk about the next one, that is ground tissue system, it includes collenchyma, sclerenchyma, parenchyma and mesophyll cells. The last one we have is vascular tissue system, which includes 3 things, that is xylem, phloem and cambium. Xylem, phloem and cambium are the tissues which are included in vascular tissue system. So with this we have completed solution number 9. Now let's discuss question number 10 on the screen. That is, how is the study of plant anatomy useful to us? Right, so we have so many advantages of plant anatomy. As you can see on the screen, we have already written the answer of this question. Plant anatomy is useful. We are going to write this answer like this in our board exams. So let's have a look. Plant anatomy is useful to us in so many manners, in so many ways. If we talk about some of them are mentioned on the screen. That is, it is helpful in understanding the structural adaptations of plants towards their diverse environmental conditions, right? Plant anatomy is very useful to understand structural adaptations of plants towards their diverse environmental conditions. Also, it helps to distinguish between monocots, dicots and gymnosperms. Along with these, also this plant anatomy is useful or helpful in improvement of food crops. Then they also can be helpful to predict the strength of wood so that we can use this wood up to its maximum potential. Last but not least, this plant anatomy also helps to understand the commercial exploitation of some plant fibers such as jute, flax, etc. Right? So this is the solution of question number 10. So let's talk about the next question, that is question number 11, which is the second last question of this chapter. And this question is asking, what is periderm? How does periderm formation takes place in the dicot stems? So we have asked about what is periderm and how the periderm formation takes place in dicot stems. So first of all, periderm is the component of secondary growth, and secondary growth always found in dicot stems and not in monocot stems. So first of all, what is periderm? Periderm is the component of secondary growth in dicot stems, and the periderm is actually which is a component of secondary growth. So it is found on the surface of root and stems of dicot plants. Periderm being a component of secondary growth in dicot stems, they are found on the surface of stem and roots of dicot plants. If we talk about the components of components or the structures included in the periderm, then periderm is composed of phallogen, phallem and phalloderm. Now what happens, or how does the formation of periderm takes place? Let's discuss this. So first of all, if we talk about phallogen, phallogen is also known as cork cambium. The cells of cork cambium or phallogen shows bipolar division. That means it cuts off the cells on either sides, that is on the outer side and on the inner side. The cork cambium or phallogen forms the cells on the outer side are known as phallem, which are suberized and have some depositions of tannins and are dead cells, which is known as phallem, of course. Now the cells which are formed on the inner side by phallogen are known as phalloderm or secondary cortex. The outside cells or the phallem cells or the layer of phallem have some aerating pores in it at some places, which is known as lenticels, and these lenticels helps in gaseous exchange but are impervious to water. So this is solution or explanation of question number 11. Let's write answer for this. So first of all, we are going to write what is periderm. So periderm is the component of secondary growth, which is found, secondary growth which is found on the surface of stem and roots of dicot stem. Now this periderm consists of phallogen, phallem and phalloderm Now we are going to talk about how the formation of this periderm takes place. So what happens? This phallogen, which is also known as cork cambium, shows bipolar division, That means this cuts off the cells on either sides and forms. This forms the layer of cells on outer side known as phallon, and this consists of aerating pores called as lenticels, which is responsible for gaseous exchange. It also, Phallogen also forms a layer on outside. So phallogen also forms a layer on the inner side and is known as phallodome. So all these phallodome, phallum and phallogen constitute together to form a new layer known as periderm, which is a component of secondary growth in dicot stems and root, right? And this is how it actually forms, or the formation of periderm takes place. So with this we have completed question number 11. Now moving on to the last question of this chapter, that is, describe the internal structure of a dorsiventral leaf with the help of a labeled diagram. So in this question we have to describe the internal structure of a dorsiventral leaf along with the labeled diagram of it. So first of all, to answer this question we have to know that in which kind of plants dorsiventral leaf is found. So clearly we know that dorsiventral leaves are present in dicot plants, right? Dorsiventral leaves are present in dicot plants. Now basically, if we talk about the description of internal structures of these dorsiventral leaves, then they have three parts, or I may say that these dorsiventral leaves are made up of or composed of three main parts, that is epidermal layer, mesophyll and vascular bundles. If we talk about epidermis or epidermal layers of dorsiventral leaf, then the outermost covering of these leaves are epidermis, and we have two types of epidermis in the dorsiventral leaves, that is adaxial epidermis and abaxial epidermis. Suppose this is a leaf and outer portion or outermost covering of a leaf is known as adaxial epidermis, whereas the lower part or the lower covering of a dorsiventral leaf is known as abaxial epidermis. Now in the dorsiventral leaf they have a thick waxy layer which is known as cuticle. Also they have stomata, but stomatas are higher on the abaxial epidermis, whereas they can be very few or less in number or may be absent on the adaxial epidermis. This is about epidermis. Let's move on to the next part, that is mesophyll. Now these dorsiventral leaves have mesophyll cells, and these mesophyll cells are of two types, that is palisade mesophylls and spongy mesophyll cells. These palisade mesophyll layer is present below the epidermis, below the adaxial epidermis, and we have another type of mesophyll cells, that is spongy mesophyll cells, which is present below the palisade mesophyll cells and above the abaxial epidermis layer. The spongy mesophyll cells also have intercellular spaces. Now let's move on to the last part, that is vascular bundle. Then vascular bundles are present in dorsiventral leaves. Yes, they are present but are surrounded by bundle sheath cells. Right? So this is all about the description of internal structure of dorsiventral leaf. Now let's write answer for this last question, and it will go like this. First of all, this dorsiventral leaf are present. Leaf is leaves. If we are writing leaf, then we are going to write is. Dorsiventral leaf is present in dicot plants. You will find the dorsiventral leaves in dicot plants only. Now let's discuss the internal structure. These leaves have three components, three main components. Such as, if we talk about that, this is epidermis. So they have two epidermis are present in them. Two epidermis are present. One is adaxial epidermis. Adaxial epidermis on upper side, on the upper side, and abaxial epidermis on the lower side, on the lower side. Now this abaxial epidermis have higher number of, this abaxial epidermis have higher number of stomata, as compared to adaxial epidermis. Now let's come towards the second thing, that is mesophyll cells. Mesophyll. So basically we have two types of mesophyll cells. First one we have is palisade mesophills, present below adaxial epidermis. The second one we have is spongy mesophyll, present below palisade one and they are having intercellular spaces. The last one we have is vascular bundles. So these are surrounded by bundle sheath cells and can be seen in veins and midribs of leaves. Vein and midribs of leaves. So this is the description of internal structure of dorsiventral leaf. Now we are going to draw the diagram of this leaf. So let's try to write or draw the diagram. This is your first adaxial epidermis. This is adaxial epidermis. Then we have palisade. We have palisade mesophyll cells like this, which are just below adaxial epidermis. Here we have vascular bundles, and vascular bundles are surrounded by bundle sheath cells. Then also we have spongy mesophyll cells like here. These are spongy mesophyll cells. Then we have abaxial. Then we have abaxial epidermis like this. We have abaxial epidermis. So this is the complete diagram of dorsiventral leaf, the anatomy of dorsiventral leaf. Let's try to label this. First of all, if we talk about, this is adaxial epidermis. These are palisade mesophyll. Then these are spongy mesophills. These are spongy mesophills. Then this is vascular bundle. I'm writing it as VB. These are bundle sheath cells. In the light yellow color, these are bundle sheath cells. This is abaxial epidermis. So with this we have completed the diagram of internal structure of dorsiventral leaf, and we have completed the solution of question number 12. With this we have completed the whole chapter of this NCERT discussion, that is anatomy of flowering plants. So this is all for today. Thank you and have a nice day.